This is the video explanation for my CSC 231 quarter project. My MATLAB script is a DNA sequencer which aims to help amateur and professional researchers to quickly analyze genetic sequences by comparing them to find the mutations and the locations within the script that these mutations occur at. This has many possible applications. On the small scale, students at Cal Poly, most likely biology and biomedical engineering students, could use this to speed up their research. It could also expand to help other majors and universities. It could also be used by professional researchers to make the process of finding a mutation faster and in some applications have the potential to help save lives. So if we look at the script that the users would initially see here, you can see it's very simple and easy to navigate. I have named it the DNA sequence tester, and it consists of just two inputs, X and Y, and a line of code that uses the function that I will explain in a moment. However, if we just run the script with the test sequences that I have inputted already for the purpose of this video, we can get an idea of what the script does. The first output is the location output, so we see here that there is a mutation in the second, third, and fourth indexes. What this means is the second, third, and fourth places in both sequence strings are different. Then below, we see the mutation output, which outputs a character array that shows what the mutations are. So in the second index, we see that the mutation is G in the X input and T in the Y input. There are two other mutations, TG and CT, at the third and fourth indexes respectively. So how does this work exactly? In the function definition in the first line, we see that this function called DNA has two vector inputs X and Y and outputs two vectors mut and N, which correlate to mutation and location. The help comments explain this as well. The first section of code sets mut and loc to empty vectors. This allows the loop to add the incremented data necessary to an empty vector. We start at n equals 1, which is the first index in both the X and Y inputs, and we set m to the length of X, which as I'll explain later is the same as the length of Y. For our while loop, we increment while n is less than or equal to the length of X, which basically means that we are incrementing through the entire input vector. Within the loop, we have some if-else statements, so if the value at X value at a specific index of X is not equal to the value at that same index in Y, then we add those X and Y values into a column vector in the mutation character array. We also add that index to the empty location vector. Then we end the if-else statements, increment by 1 to the next index value, and then end the while loop. After the while loop, we display these location and mutation vectors as well. So going back to the script, you can see I've written some comments to help the users navigate and troubleshoot if necessary. The first states that the inputs must be strings since these are characters, not numerical values. This just means that the user must make sure that there are apostrophes at the beginning and end of their sequences that they input. The second states that it is important that there are no spaces in the string. This is so the location vector is correct because spaces in a string take their own index. The third comment states that the sequences must be the same length. So now we're going to pretend that we're a potential user so we can see how the script is used from an outside perspective. I have been using a website created by UC Riverside that is a random sequence generator. So for the purpose of this video, we will use this site to create two random sequences. To keep within reasonable time limits, we're just going to analyze two 4 base pair sequences. So we're just going to change this to 4 for the amount of base pairs and generate our first EDAC. And then we're going to copy and paste that into the first input and create a second ACAC. And copy and paste that to the second. So as you can see, we have two mutations in the first and the second indexes, a G and an A and a G and a C. Then we have an A and an A and a C and a C. And since those are the same, they won't show up in our output. So we're just going to run it and see what we get. And like we thought, we have mutations in the first and second index, like we said, with the G and the A and the G and the C. And then our mutation vector gives us those values. As you can see, the script is very simple and user-friendly, and I really hope that it can be useful to many other people.